Hi, everybody. My name is Matthew. I'm a PhD student working with Dr. Charles Ofria at MSU, and today I'm going to be talking to you about our work developing digital models of multicellularity. Our work is really motivated by foundational questions in biology about the evolution of complexity and diversity on Earth. In foundational theoretical work on major transitions in evolution, Smith and Smarthy make a connection between the emergence of new replicating entities through the evolutionary process and the emergence of complexity and diversity. And one key type of evolutionary transition in individuality that Smith and Smarthy highlight in their work is the evolution of multicellularity. I've got an example up on the slide from Will Ratcliffe's lab, where he used experimental evolution in a wet lab to go from unicellular yeast on the left here to multicellular yeast strains on the right. There's also been some really exciting work in the digital evolution space, looking at the evolution of multicellularity, notably Heather Goldsby's theme-based work. And what we're really interested in doing is kind of extending this work to allow for more nuanced interactions between multicellular individuals to introduce kind of an element of biotic selection into the mix, which is thought to also be potentially related to complexity and diversity. And so in our system, cells interact in a unified spatial realm. What we have here, individual little tiles represent individual cells that then grow and proliferate through cellular reproduction, forming color-coded multicellular groups that then interact freely within a unified spatial realm and can, via their constituent cells, do arbitrary message passing, resource sharing, and compete for space. And this allows for kind of free-form interactions between multicellular groups. For example, multicellular groups can provide resource and protection to their offspring groups. And so far, a lot of our work with the system has been characterizing the types of phenotypes that we see evolve in it. We've seen some hallmark characteristics of multicellularity evolve, such as reproductive division of labor, apoptosis, and resource sharing. To give you a sense of what that corresponds to in our system, one of the common types of reproductive division of labor that we see evolve is differentiation between interior and exterior cells, where interior cells have a hugely depressed cellular reproduction rate, where they're kind of becoming quiescent, and most of the cellular reproduction is focused on the exterior, the periphery of groups, where there's kind of more competition for space with other groups. And a lot of the work that we've been doing has been kind of characterizing individual case studies to get a sense for what's evolving. I'm going to share 2 case studies with you. And I've got links on the bottom to live in-browser simulation where you can see each of these strains in your own web browser, and it's mobile friendly, so be sure to give that a try. All right, so what I'm going to show you here is a time series with a parent group colored in purple, and we can see the strain seeds endogenous propagule groups, one of which is colored in orange, that then grow over time, eventually destroying the parent, but yielding several viable offspring groups. And so we were really interested if there were cellular mechanisms that were underpinning this interior propagule generation. And so we looked at gene regulation, which I've got in a color-coded visualization on the left, showing kind of the spatial distribution of gene regulation patterns within these multicells. And we disable gene regulation to create a knockout strain, and then we assessed that knockout strain in terms of its relative fitness and the incidence of propagules, and we observed lower relative fitness and a lower incidence of propagules, kind of suggesting that this interior propagule generation strategy is being mediated by cellular processes, and that's adaptive. Another thing we've been investigating with the system is kind of this ability to select for more holistic functional integration across groups. And so in a kind of a variant of our model, we introduced the ability for groups to grow long distance interconnects between cells via a development-based search process that you can see on the right, and yielding as kind of established interconnects after that development process concludes, which you can see on the left here. And we've seen cells use these kind of long distance interactions with other cells adaptively. In one case study, we saw cells at the periphery of groups were sending messages over interconnects that would then trigger the cells that receive those messages to send resource the other way. So the net effect here was basically resource being pumped from the interior of groups to the exterior of groups. And we validated that the functionality of the system by doing knockouts of individual components, and with each component we saw a decrease in relative fitness when we knocked it out. So just to wrap up here, in the continuing work, we're really interested in exploring metrics to assess the evolution of complexity and looking at questions about how the evolution of complexity relates to fitness. And with that, I'd like to thank my collaborators first and foremost, my advisor, Charles Ofria, Alex Lolagini, a grad student who's been really involved in a lot of the Signal GP stuff and helping to integrate that into our system. I'd like to thank the undergrads I've had the chance to work with, Nathan and Santiago. Nathan, who kind of helped build some of the computational infrastructure that we're using in the simulations, and Santiago has been doing a lot of work on our web visualizer, which I've got linked again on the bottom of the slide. And with that, I'll take questions. Thank you.